The main problem for secondary progressive MS has been that it's a little bit of a slower progressive problem. When you look at a relapsing population of patients, you can measure attacks. And one of the outcomes for many of our clinical trials is attack rate. So you can measure that. People come in, they have new symptoms. You can actually say, oh, you've reached this level of EDSS abnormality on the scoring, so you've had an attack. The problem we have with secondary progressive MS is that there is not as easily defined events. Early in the course of secondary progressive disease, you can still have attacks, but as the disease progresses, you may no longer have attacks, and you just slowly get worse over time. And when I mean slowly, the change, our ability to measure that change on our EDSS scoring is very difficult because the change can be over years. So a small inability to go from a normal gait to maybe requiring a cane may take years. And we design clinical trials sometimes with the idea that you want to measure something in 2 years, possibly 3 in some trials. And the problem with that is that most patients may not progress that much in that time. So if you're going to take a group of patients and put half of them on placebo and the other half on an active treatment, you may have a difficult time in a short amount of time to demonstrate a meaningful difference between these two groups. That has been a huge problem for the MS community, dealing with progressive patients, period. And that's both primary progressive and secondary progressive. But that metric of measuring disability on this EDSS scoring system is not sensitive enough to those changes over time. So that's been the big problem. Most recently, we've been doing a clinical trial with a molecule called siponimod, which is very similar to fingolimod. And fingolimod is the drug that's approved for relapsing disease. And it's a S1P modulator. It's an agonist. It binds to a receptor that is present on your lymphoid cells, and it causes internalization of the receptor. And that receptor is actually required for lymphocytes to migrate out of lymph nodes to get into the blood circulation and go do their thing. So when you look at the fingolimod molecule, it hits many different receptors. There are 5 receptors, subtypes, and it hits all of those. And we believe that the side effect profile for fingolimod is in part because those receptors are so ubiquitous through the body. It's on your lungs, it's on your heart, it's on pretty much most tissues of your body. And that is why fingolimod has a lot more requirements for monitoring. You have to have cardiac monitoring. You need to have your eyes checked. You may need to have liver tests, lung issues, pulmonary function tests. So there are a number of things that go on with that compound. When we thought about the idea of doing a trial for a more selective molecule, one that only binds to the S1P1 receptor and the S1P5 receptors, you might be able to get rid of some of the other side effects with this drug. And so there's been a number of companies out there trying to develop compounds that are more selective to be able to get you efficacy but not require all of that monitoring and safety concerns. So siponimod was one of these. And they've done the trials, and they did a trial in secondary progressive MS. And the way this study was designed was the idea that they could look at slowly progressive disease, secondary progressive patients who met certain entry criteria. They needed to have a certain level of neurologic disability. And 2/3 of them would go on to active treatment, 1/3 would be on the placebo group, and they would go ahead and do a 2-year trial and try and see how these patients did. But 2 years may not be enough to be able to answer the question. So they devised the trial to make it an event-driven trial. And what that means is that you need to have a certain number of events of people progressing in the trial before you could say this trial was complete. So they completed the trial, and the data was impressive because this is the first compound that was able to show a benefit in secondary progressive disease. And it showed about a 21% reduction in disability progression. It hit on all the other metrics of relapses for people who are still having relapses, MRI activity. So it looked very similar to what you got with fingolimod in terms of the benefit, but it also hit the disability progression piece that you were not able to see in other trials in secondary progressive patients. So this is really the first time that we've seen this kind of a benefit.